Structure of viruses. A virus is a small infectious agent that can only replicate inside the cells of another organism. Viruses of all shapes and sizes consist of nucleic acid core, an outer protein coating known as capsid, and sometimes an outer envelope. The virus core contains the single or double-stranded genome, RNA or DNA, that encodes the viral proteins that are required for its replication. The capsid and its enclosed nucleic acid together constitute the nucleocapsid. Capsid is a protein that surrounds the nucleic acid of a virus. These capsids are responsible for different symmetries of viruses. Capsomere is a structural unit of a viral capsid. These self-assemble in different ways to form either icosahedral symmetry, helical symmetry, or complex symmetry. The basic subunit which makes the capsid is called as protomer. First is the icosahedral symmetry. Viruses having icosahedral symmetry appear spherical in shape, but a closer look actually reveals their real symmetry. It is a polygon with 12 vertices, 20 faces, and 30 edges. Each face is a shape of an equilateral triangle. Nucleic acid is deeply situated and protected by capsid. This symmetry is also called as 2:3:5 fold rotational symmetry. Example: in capsomere of adenovirus, on edges and faces, capsomere bond to 6 neighboring capsomeres called as hexon, and at vertices bonds to 5 neighboring capsomeres called as penton. This is the example of adenovirus. Second is the helical symmetry. These viruses are composed of a single type of capsomere stacked around a central axis to form a helical structure. Capsomere and nucleic acid are bound together to form a helical or spherical tube, which may have a central cavity. This symmetry has identical protomers as its subunit. Morphological subunits are arranged in their long axis in a nucleocapsid in a screw-like manner in several units per turn of a helix. In animal RNA viruses, helical nucleocapsid is always inside the glycoprotein envelope to give more stability, but in plant viruses it is non-enveloped. This arrangement results in broad shape of filamentous virions. Example: enveloped animal RNA virus family with helical symmetries Rhabdoviridae, and non-enveloped plant RNA virus family with helical symmetry is tobacco mosaic virus. Next is complex symmetry. These viruses possess a capsid that is neither truly helical nor truly icosahedral, and that may possess extra structures such as protein tails or a complex outer wall. These virus structures may have a combination of icosahedral and helical shape, may have a complex outer wall or head-tail morphology. Few viruses like pox virus, retroviruses, and bacteriophage have complex symmetry. Example is pox virus have tumble-shaped core surrounded by complex membrane and is slightly pleomorphic ranging from ovoid to brick shape. Retrovirus has tubular nucleocapsid surrounded by icosahedral capsid enclosed within envelope. Envelope. This virus structure is a conventional icosahedral or helical structure that is surrounded by a lipid bilayer membrane. Enveloped virion acquire their outer layer when nucleocapsid is extruded through one of the cellular membrane in a process known as budding. Lipids are derived from host cell membrane and proteins are virus coated. Envelope associated proteins are associated with at least 4 crucial activities, that is, receptor binding, membrane fusion, uncoating, receptor destruction. Glycoproteins, usually in the form of dimer, trimer, or tetramer, assemble into the virion papillomas or spike, as seen on rhabdovirus, orthomyxoviruses, paramyxoviruses, phyllocorona, arena, and retroviruses. Fusion proteins are glycosylated and are also associated with papillomas involved in the viral entry and viral release. Matrix proteins are non-glycosylated and are found as a layer on the inside of the envelope of orthomyxo, paramyxo, rhabdo, phyllo, and retroviruses. Matrix proteins provide added rigidity to these viruses. The infectivity of most envelope viruses depends on the integrity of the envelope. However, because the envelope contains lipids, it makes the virus more susceptible to inactivation by environmental agents such as detergents that disrupt lipids. Significance of understanding the virus structure is to classify new viral isolate for confirmed diagnosis of virus infection and studying the pathogenesis or infectivity of a virus. Thanks for watching. Please subscribe to channel.